Hello and welcome to another episode of Medicine Can Be Easy. Today we are going to continue with the physiology of the cell. Last time we talked about the cell structure, membranes, endoplasmic reticulum, and Golgi apparatus. If you haven't seen the video, then the link is in the description. The last thing we said was that the Golgi apparatus forms lysosomes, secretory vesicles, and other cytoplasmic components. So let's take a look. We'll start with lysosomes. Lysosomes are vesicular organelles that are responsible for digestion within a cell. The cell can use them to digest damaged cellular structures, ingested particles, or bacteria, for example. Lysosomes are 250 to 750 nanometers in diameter. For comparison, we said that the cell membrane is 7.5 to 10 nanometers thick. They have the usual lipid bilayer membrane, and they contain small granules with digestive enzymes, the so-called hydrolase enzymes. By using these enzymes, they are able to split an organic compound into two or more parts. As we said, the cell uses lysosomes for digestion. First, the matter that is to be digested must be brought into the cell by phagocytes, special cells that surround and engulf it. Then they transport it through the cell membrane. The membrane of the lysosome fuses with the membrane that surrounds the phagocytized material, and this enables the lysosomal enzymes to digest it. Now let's talk about peroxisomes. Peroxisomes also have a membrane, and they contain several specific enzymes called oxidases. Oxidases are enzymes that are capable of reducing oxygen to hydrogen peroxide and hydrogen peroxide to water. They are also able to oxidize many substances that would otherwise be poisonous, like, for example, alcohol. Peroxisomes are not formed by the Golgi apparatus, but they self-replicate. They are particularly large and active in the liver. Then we have secretory vesicles that are released from the Golgi apparatus into the cytoplasm as storage vesicles. The cell uses them for secretion of special chemical substances. Next we have mitochondria, the powerhouse of the cell. Mitochondria serve as sites for the production of the energy of cells. They self-replicate and contain their own DNA. They have two lipid bilayer protein membranes, an outer membrane and an inner membrane. The outer mitochondrial membrane is smooth, but the inner membrane is characterized by many folds called cristae. The inner cavity of the mitochondria is filled with matrix, which contains dissolved enzymes that are used for extracting energy from nutrients. The folds in the inner membrane, the cristae, contain oxidative enzymes. The enzymes in the matrix cooperate with these oxidative enzymes, and they do two things. They oxidate nutrients and at the same time release energy. And the energy is used to synthesize adenosine triphosphate, the enzyme that fuels many cellular activities. Now let's take a look at the nucleus. The nucleus is the organelle that contains the DNA of a cell. It has its own membrane, the nuclear membrane, which consists of two separate bilayer membranes. The outer membrane, which is continuous with the endoplasmic reticulum, and the inner membrane, that together form the nuclear envelope. It's penetrated by several thousand nuclear pores through which molecules pass. Nucleus is important for cell growth, metabolism, and reproduction. It contains chromosomes that control cellular activity and protein synthesis. A nucleus may contain one or more nucleoli. The nucleolus doesn't have a membrane. It consists of large amounts of RNA and proteins that are similar to those that are found in ribosomes. The last thing we'll talk about is the cell cytoskeleton. First, we need to mention fibrillar proteins. Fibrillar proteins in the cell are usually organized into filaments and tubules. Filaments come from precursor protein molecules that are synthesized by ribosomes. When they polymerize, they form filaments. For example, you probably know actin filaments that form the basis for muscle contraction. A different type of filaments is composed of polymerized tubulin molecules that form strong tubular structures, microtubules. Microtubules provide physical structures for certain parts of the cell. Microtubules and microfilaments together form the cytoskeleton. It's a structure that is capable of rapid movement and reorganization, so even though it's called cytoskeleton, you shouldn't imagine it as something very rigid. One of the functions of the cytoskeleton is to provide a system that different organelles and molecules can use to travel. There are three main proteins responsible for this function: myosin, which moves along actin filaments, and kinesin and dynein, which move along the microtubules. The way this works is that one end of the protein attaches to the molecule that is to be transported, and the other end moves along the microfilament or microtubule. Okay? Next time, we'll look at the different functions the cell is capable of, so stay tuned. If you like this video, please subscribe to learn more about the cell, physiology, and much more. You can also follow us on Instagram, where we post multiple choice questions that you can use to practice what you've learned. Thanks again, and see you next time.